Good afternoon. In this session, we are going to continue our discussion of cellular respiration, and we are going to discuss the properties of glycolysis. We have discussed several properties in the previous class, and today we are going to continue discussion of the properties of glycolysis. As we said, the ATP that has been formed in glycolysis is formed by substrate level phosphorylation. This is one type of direct phosphorylation in which a phosphate group is added directly to the ADP molecule. So let me use the pen, please. Okay, so if we have ADP and you add a phosphate group and of course energy, because this is the purpose of that endothermic reaction, then you're going to store energy in this molecule, which is called ATP. This is an example of substrate level phosphorylation where there is direct transfer of phosphate to ADP. Since ADP contains only 2 phosphate groups, then ATP will contain 3 phosphate groups. This direct substrate phosphorylation is called direct because it does not need the molecule to enter the mitochondrion. It can occur in the cytosol, and of course, it requires a phosphorylase enzyme or an enzyme that phosphorylates ADP. But the problem with substrate level phosphorylation is that it produces only a small amount of ATP if compared to the amount of ATP that is formed from the breakdown of one molecule, the complete breakdown of one molecule of glucose. In number 11, we discuss the enzyme that is called phosphofructokinase. Of course, as its name indicates, it is a kinase enzyme, meaning that this enzyme will break down an ATP molecule to convert the ATP into ADP plus inorganic phosphate plus energy, and then this phosphate will be used to phosphorylate an already phosphorylated substrate, which is fructose 6-phosphate. Since fructose, the substrate in this case, already contains a phosphate group, then we call it fructose 6-phosphate. So phosphofructokinase will phosphorylate an already phosphorylated fructose. So at the beginning, fructose had only 1 phosphate group, and at the end of the reaction, it will contain 2 phosphate groups, as we will see on the next slide. Phosphofructokinase enzyme is an allosteric enzyme, and as we have studied before, allosteric enzymes are enzymes that have 2 important sites. They have an original site, which is the active site that is specific to the substrate, which is here fructose 6-phosphate, and they have another site, which is called an allosteric site. This allosteric site is capable of binding with certain molecules that can bind here and that are called allosteric effectors. And when the allosteric effectors bind with the allosteric site, they cause a conformational change in the shape of the enzyme. That conformational change may have a positive effect on the active site, meaning that it helps the active site to bind more perfectly with the substrate, or it might have a negative effect on the active site and therefore on the whole enzyme by making the active site less fitting with the substrate. So if the allosteric effector results in improving the function of the enzyme, then it would be an allosteric stimulator. How would I know that it is an allosteric stimulator? First of all, because binding with the active site would occur more perfectly, and second, by an increase in the rate of product formation by the reaction. If, on the other hand, the allosteric effector results in conformational changes in the enzyme, that will result, of course, in conformational changes in the active site. And those conformational changes in the active site result in diminished rate of binding with the substrate, and therefore a diminished rate of production of the products, then it would be an allosteric inhibitor. And we will see how this allosteric property of phosphofructokinase is important for regulation of glycolysis and regulation of the rate of production of ATP in the cell. For example, if we look here at this diagram, here we have glucose. We start in glycolysis, of course, we start by glucose, and then we utilize one ATP molecule here to phosphorylate glucose into glucose 6-phosphate, and then we have here an isomerase enzyme, and that isomerase enzyme will convert glucose 6-phosphate into its isomer, which is fructose 6-phosphate. So since once fructose is formed, it is phosphorylated because it comes from its isomer, which was already phosphorylated, which is glucose 6-phosphate. Here we are going to consume another ATP molecule that will be broken down and hydrolyzed into an ADP molecule, and the phosphate will bind to the carbon atom number 1 of fructose, and we will have fructose 1,6-biphosphate or fructose 1,6-diphosphate. This simply means what? This simply means that the two phosphate radicals, one of them, the first one was attached to the 6th carbon atom of fructose, and the second is attached to the 1st carbon atom, meaning that if these are the 6 carbon atoms of fructose, then fructose 6-phosphate looks like this. Here we have a phosphate at position number 6, and then by further phosphorylation, we will have another phosphate at position number 1. The enzyme that stimulates that reaction is a phosphofructokinase enzyme. This phosphofructokinase enzyme stimulates the formation of fructose 1,6-biphosphate and therefore stimulates glycolysis to continue and to proceed. And the overall reaction of glycolysis is that we have a net supply of 2 ATP molecules. Not only this, but if oxygen is present, then we would proceed towards complete oxidation, towards aerobic oxidation in the mitochondrion, because glycolysis occurs in the cytosol. But if oxygen is there, we would go to the mitochondrion and further break down the carbon skeleton of glucose and get or release the stored energy, and we will end up by having at least 36 ATP molecules. So glycolysis is crucial in aerobic cellular respiration. Although glycolysis itself is an anaerobic process, it does not require oxygen, or it does not necessitate the presence of oxygen. We don't use the oxygen molecule in the cytosol to carry out glycolysis. But the problem is, if you stop glycolysis, then the whole procedure afterwards will be stopped. So if you stop that enzyme that is phosphofructokinase, then you would stop the process of aerobic cellular respiration and therefore stop the production of energy. Phosphofructokinase is an allosteric enzyme, meaning that in addition to the active site, which binds specifically to the substrate, which is fructose 6-phosphate at the active site. This is actually the active site. There is another site which is called the allosteric site, and that allosteric site can bind with the ATP molecule. Okay, if you have plenty of ATP in the cell, then if you have plenty of ATP in the cell, then ATP molecules would bind with that allosteric site, the red one, of the enzyme that is called phosphofructokinase. This is the name of the enzyme, and will cause conformational changes. And those conformational changes will have a negative effect on the active site, making it no more capable of binding with the fructose 6-phosphate, okay? And this simply means what? This simply means that if you have plenty of ATP in the cell, you will stop the action of phosphofructokinase or diminish the action of phosphofructokinase, and therefore diminish or stop glycolysis. And since glycolysis leads to Krebs cycle and the electron transport chain, then you will diminish Krebs cycle, you will diminish the electron transport chain, and therefore you will diminish energy production by the cell. This is a kind of auto-regulation, and we call that kind of auto-regulation feedback inhibition. What do we mean by feedback inhibition? Whenever you have feedback inhibition, this is what we call, another name for it is what we call negative feedback or a negative feedback loop. A negative feedback loop. What do we mean by a negative feedback loop? Here we have the initial substrate, which is phosphofructokinase, and here is the biochemical pathway. You have intermediate A, meaning the first product, and then intermediate B, and so on. This is just a symbolic diagram. It does not exactly give you the number of chemical reactions, but actually here we have an end product. Our end product at the level of glycolysis, at the end of glycolysis, you get ATP. This end product can also be called an output, okay? This output will have a negative effect on one of the reactions of the, one of the first reactions of glycolysis. So here you have the output has a negative reaction on the input, or more accurately speaking, part of the output inhibits the input. And this is what we call negative feedback inhibition. Negative feedback inhibition does not only occur in glycolysis, but it occurs in many biochemical reactions in the human body, like, for example, in hormonal secretion, for example. So whenever a gland secretes hormones and the level of the hormone increases, then there would be mostly a negative feedback loop to stabilize the level of the hormone in the blood. If the gland continues secretion without a negative feedback inhibition, then the levels of the hormone would exceed the required levels, and this would be harmful to the human body. So the main purpose of negative feedback loops is homeostasis. This example of feedback inhibition of, here we have feedback inhibition, here we have a feedback inhibition of phosphofructokinase enzyme by what? By the output, which is the ATP. Okay, so this is one important thing to understand, and this is an answer to the question, how does the cell, we usually don't ask you how does the cell equilibrate or control the amount of ATP that is produced, but sorry, the reverse. We usually don't ask you how we regulate glycolysis or how does feedback inhibition occur, but we ask you this question at the bottom of the slide, which is how the cell regulates ATP production. And here we have seen how the cell regulates ATP production by feedback inhibition. In this important diagram, we see how we shift from glycolysis to, which occurs in the cytosol, to the inside of the mitochondrion, where we will have Krebs cycle, and followed by, so in the cytosol, we have glycolysis. And as we said, glycolysis is a common pathway between aerobic and anaerobic respiration, and glycolysis per se does not require oxygen. And if oxygen is not available, then the end product of, or one of the end products of glycolysis, which is pyruvate, will stay in the cytosol and undergo reduction into lactic acid or ethyl alcohol and carbon dioxide, in which case the whole process of glycolysis followed by pyruvate reduction would be called fermentation, acidic fermentation or alcoholic fermentation. But in case of availability of oxygen in the mitochondrion, and as we said, oxygen is the final electron acceptor in the electron transport chain, then the whole process will proceed. So pyruvic acid in such a case would need to enter the mitochondrion. This is the formula of pyruvic acid. As you see, it is a 3-carbon compound, and it is CH3, C double bond O, C double bond OOH. So here we have a carboxylic group. Pyruvic acid loses in the aqueous solution of the cytosol, pyruvic acid loses a hydrogen atom and the oxygen becomes negatively charged. So since the pyruvate molecule is charged, then it is hydrophilic, then it cannot diffuse easily through the mitochondrial membranes. It has to diffuse through the outer mitochondrial membrane and then through the inner mitochondrial membrane. And here we have a transport protein that helps pyruvic acid to diffuse through the mitochondrial membrane and enter in the matrix of the mitochondria. What happens actually is that before passing through the inner mitochondrial membrane, pyruvic acid stays here. So the transport protein first allows pyruvic acid to pass through the outer mitochondrial membrane, and then pyruvic acid stays here in what we call the outer mitochondrial space. So when we say outer mitochondrial space, we mean the space between the outer mitochondrial membrane and the inner mitochondrial membrane. It's a bit confusing because we say outer space, but the outer mitochondrial space is the space between the two, and we refer to it as OMM, outer mitochondrial space. And then in order for pyruvic acid to pass or diffuse through the inner mitochondrial membrane, there is a shuttle molecule that is called coenzyme A. That coenzyme A will help pyruvic acid to pass through the inner mitochondrial membrane. Not only this, but it will activate pyruvic acid in order to start the reactions of Krebs cycle. By the way, the reactions of Krebs cycle occur here in the matrix, and the reactions of Krebs cycle will be followed by the electron transfer chain. This is the third and last step of cellular respiration, and the electron transfer chain occurs on the inner mitochondrial membrane. Okay, so here in this reaction, before starting Krebs cycle, pyruvic acid undergoes a very important chemical reaction, which is called oxidative decarboxylation. Oxidative decarboxylation. What is oxidative decarboxylation? As its name indicates, it is decarboxylation associated with oxidation. Where is the decarboxylation here? Let's look at the molecule of pyruvic acid to the left and the molecule of pyruvic acid to the right, but first let me erase all the editing. So here, let's look at the molecule of pyruvic acid. If you remove all this, if you remove the carbon dioxide molecule, then you will only have CH3C double bond O, which is the acetyl group. Of course, I'm oversimplifying things. It's not exactly like this, but this is the overall reaction. It is as if you removed CO2, which is carbon dioxide, from the reaction. So what remains here is the acetyl group, which is CH3C double bond O. That is the acetyl group. Acetyl group. So there is actually a molecule of carbon dioxide that is removed. That's why we call it a decarboxylation reaction, a decarboxylation reaction. And then there is binding of this shuttle molecule, which is coenzyme A, with the acetyl group to form acetyl coenzyme A. Acetyl coenzyme A. And here, coenzyme A ends by SH, okay? So there is an H that is removed from here, another H that is removed from the acetyl radical. So we have two H's that are removed, and these are carried by NAD, as we saw last time in the classroom. As I said, this is the, for those who were absent, this is the oxidized form of NAD, nicotinamide adenine dinucleotide. It's called a nucleotide because it's similar to the nucleotides in DNA, because it contains adenine. And nicotinamide is a member of the vitamin B complex. Here, the positive means that it has lost an electron, and when it gains two hydrogen atoms, they are being gained like this. This is the proton, and this is the electron. So this is how we represent the two hydrogen atoms. This electron will neutralize NAD, and therefore NAD will become neutrally charged, and this whole H will bind, and then the other one, which is only a proton, will be alone. So as NAD moves in the mitochondrion, it's like this. It's reduced NAD having one hydrogen atom bonded to it, and another proton that's all the time accompanying it as it moves along the inner mitochondrial membrane or inside the mitochondrial matrix, okay? So what's the function of coenzyme A? The function of coenzyme A, number one, is to transfer pyruvic acid. This is not written in your book, but it's good to know that it helps it move through the inner mitochondrial membrane, and then it activates it. And then it activates the acetyl group to join the acetyl group that resulted from oxidative decarboxylation of pyruvic acid to start Krebs cycle or to join Krebs cycle, okay? Why does it activate it? Because we have a high energy bond here, a high energy bond between coenzyme A and the S, okay? Now let's move on to the next concept here. Of course, this is how allosteric inhibition of phosphofructokinase occurs. And here, this is allosteric activation. This is the allosteric effector. We said that the general name is an allosteric effector. If that allosteric effector helps further or more perfect binding of the substrate and makes the enzyme have the correct conformation for the substrate, then that allosteric effector is an activator. But if the binding of the molecule here resulted in a conformational change that is a wrong or incorrect one, just because the substrate is now no more capable of binding with the active site, then it would be an allosteric inhibitor. And of course, the best example here would be ATP, that is an allosteric inhibitor of phosphofructokinase. And this is the concept that is delivered by this slide, that ATP is the perfect allosteric inhibitor for phosphofructokinase, and that the result of this is regulation of ATP production, by the cell, and the regulation of ATP production by the cell results, of course, in homeostasis. Okay, this is a good example of allosteric regulation. This is a good example of regulation of ATP production by the cell, and this is also a good example of regulation or homeostasis in the body. We said, of course, that when oxygen is present, we are allowed to carry out aerobic respiration, and that aerobic respiration is highly efficient because per glucose molecule, it produces more ATP, more than what? More than glycolysis and more than aerobic respiration in general. Because we will see that in fermentation, for example, the two ATPs that have been, we produce only two ATPs through glycolysis. Whereas in aerobic cellular respiration, it depends upon the type of organism. We produce between 36 to 38 ATP molecules. And as we said, although the process itself as a whole is called aerobic respiration, yet it consists of an anaerobic phase called glycolysis in the cytosol, and then we have an aerobic phase. The aerobic phase should be in the mitochondria. As we said last time, the aerobic phase occurs in the mitochondrion, and it should be in the presence of oxygen. And we said that oxygen is the final electron acceptor in the ETC, in the electron transport chain. So if you don't have oxygen that receives electrons at the end of the story or at the end of the day to form a water molecule, then all the electron transport molecules, protein molecules on the inner mitochondrial membrane wouldn't be able to advance the electrons, and the electrons would not release the stored energy, and ATP would not form, and the whole process would stop. The aerobic phase, so actually when we talk about glycolysis, about aerobic respiration, and when we say that it consists of two phases, we say anaerobic phase of aerobic respiration, which is glycolysis, and then we have the aerobic phase of aerobic respiration. The aerobic phase of aerobic respiration consists of the citric acid cycle and oxidative phosphorylation. But before moving on, let me go back to, or let me explain here, but first remove all the editing. Let me explain what happened to pyruvic acid. We said that what happened to pyruvic acid was called oxidative decarboxylation here. Oxidative decarboxylation. Why oxidative? Because we removed hydrogen from the pyruvic acid and we converted it into acetyl radical. So oxidative, why? Because you removed hydrogen and you converted pyruvate into acetyl. And because you removed a carbon dioxide molecule, and here the difference between pyruvate and acetate, because you removed one molecule of carbon dioxide, so definitely you will have a carbon atom less. Pyruvic acid is a C3 compound, whereas the acetyl radical is a C2 compound. Here, as you can see, we have only two carbon atoms. So it is oxidative because we removed two hydrogen atoms that were carried by NAD, and it is decarboxylation because we removed carbon dioxide. So here it is decarboxylation. And I repeat again that oxidative decarboxylation means decarboxylation associated with oxidation or removal of carbon dioxide associated with removal of hydrogen. So let's open a screen and say here oxidative decarboxylation, oxidative decarboxylation means what? Oxidative decarboxylation means decarboxylation that is coupled with oxidation. That is coupled with. Once you have this suffix IVE, then it is coupled with. So decarboxylation coupled with oxidation. What is decarboxylation? Decarboxylation is removal of carbon dioxide. So actually it is removal of carbon dioxide coupled with removal, here oxidation, with removal of electrons or removal of hydrogen. Okay, so it is removal of carbon dioxide coupled with removal of hydrogen. And the best example is when pyruvate is converted into acetyl coenzyme A. Okay, of course, coenzyme A is essentially SH. It contains a sulfhydryl group. Now we move on again to the citric acid cycle and oxidative phosphorylation. The citric acid cycle occurs in the matrix of the mitochondrion, and the citric acid cycle is a biochemical pathway, meaning that you have a series of chemical reactions. Every reaction leads to the following, to the next one, and the product of the first reaction is the substrate of the second reaction. So a biochemical pathway is a series of chemical reactions in which the product of a previous reaction is the substrate of the second reaction. And it occurs in the jelly-like substance inside the mitochondrion, the matrix of the mitochondrion that contains all the necessary enzymes for the reactions of Krebs cycle to be activated. Of course, we need pyruvic acid that results from glycolysis, and therefore glycolysis should always precede a Krebs cycle, and Krebs cycle completes or shares actually in completing the oxidation of glucose to carbon dioxide. What does this mean? This means that if this is the carbon skeleton, for example, and you break down that carbon, so you can't have a free carbon like this, but if you add oxygen to it, then you are producing carbon dioxide. So breaking down the bonds in glucose and adding oxygen is actually formation of carbon dioxide. And that's why throughout the process of cellular respiration, throughout the process of breaking down bonds to release energy, we are actually having carbon atoms that are detached from the carbon skeleton and oxygen that is added to form carbon dioxide. And then we have the last stage. You're frequently asked about the last stage of aerobic cellular respiration, which is oxidative phosphorylation, and that is the process that occurs along the ETC. So oxidative phosphorylation, as we said before, is phosphorylation. Actually it is, let me erase. So it is phosphorylation, okay, coupled with oxidation. Again, oxidative, IVE, means coupled with. So phosphorylation coupled with oxidation, okay? Phosphorylation is let me change the color, is the addition of oxygen. So phosphorylation is the addition of phosphate. Phosphorylation is the addition of phosphate. And oxidation is either you add electrons, addition of oxygen, or removal of electrons, And oxidative phosphorylation is a process that occurs along the electron transfer chain. So again, oxidative phosphorylation is phosphorylation coupled with oxidation. Phosphorylation is addition of phosphate using enzymes, and oxidation is addition of oxygen or removal of electrons or removal of hydrogen, of course. When you remove hydrogen, you're actually removing electrons. What are the steps of Krebs cycle? Of course, you have the diagram for Krebs cycle in the book, but you're not supposed to know the details. You are only supposed to understand the events of Krebs cycle. As we said, at the end stage of glycolysis, we broke down glucose into 2 molecules of pyruvic acid. And then pyruvic acid has to enter the mitochondrion in order to do what? In order to complete oxidation of the glucose skeleton into carbon dioxide and water. Carbon dioxide and water are formed, plus an amount of energy that is necessary to form 38 ATP. So why does pyruvate enter the mitochondrion? For full oxidation of the carbon skeleton of the glucose, carbon skeleton. What do we mean by full oxidation of the carbon skeleton? We mean that we're going to break down all the bonds between the carbon atoms, and then we are going to release all the energy. That energy will be stored in ATP, and there will be oxidation, like oxidation of carbon into carbon dioxide. And meanwhile, there are hydrogen atoms that are released from the carbon skeleton. So what's the evidence of oxidation of the carbon skeleton? Meaning that what happens to glucose inside, what happens to glucose, to the glucose carbon skeleton, during aerobic respiration. The carbon skeleton, which is that skeleton we know of the hexose, 6 carbon atoms. What happens to the glucose carbon skeleton? First of all, you break down bonds. You break down the bonds between carbon atoms. And these bonds between carbon atoms, they are here to store energy. Number 2, okay, so some energy will be released. Number 2, you form carbon dioxide. So this is oxidation. So you oxidize carbon into carbon dioxide. Number 3, you remove hydrogen, and that hydrogen is carried by hydrogen carriers like NAD. And then finally, the energy will be stored in the form of ATP. So this is actually what happens to glucose. As we said, before joining Krebs cycle, there should be combination with coenzyme A. And coenzyme A is a coenzyme actually of vitamins. And then pyruvic acid undergoes oxidative decarboxylation, and at the same time, it undergoes activation. And we explained oxidative decarboxylation. And then here we have the first step, which is combining acetyl coenzyme A with a molecule that is present in the matrix of the mitochondrion called oxaloacetic acid, OAA, to produce the first product of Krebs cycle, which is citric acid, and that's why we call the whole cycle citric acid cycle. Krebs cycle has been called or named after the scientist Hans Krebs, Sir Hans Krebs. Krebs, actually, Krebs. It's not, there is no apostrophe. It's called Krebs. He is called Krebs. But because the first product in the Krebs cycle is citric acid, we call it citric acid cycle. And we will see that it is a cycle, meaning that we start at a point, the pathway proceeds, and then we end at the same point, meaning that this is the first reactant, this is the first reactant, and then you proceed in the cycle like this, and then you return again to oxaloacetic acid. So there will be, at the end of every Krebs cycle, there will be regeneration of oxaloacetic acid. Why would there be regeneration of oxaloacetic acid? To be capable of starting a new Krebs cycle again. And what's interesting to know, that for each glucose molecule to be completely oxidized, we need a Krebs cycle to turn twice. Why? Why this is important? Now, so actually I want a screen. Okay, so here in glycolysis, we have one glucose molecule, and that glucose molecule will be broken down via glycolysis that occurs in the cytosol. It will be broken down into two molecules of pyruvic acid. So you will have pyruvate here and pyruvate there. Actually, they are in the same place, but I mean that they are two pyruvic acid molecules. Why we said it before? Because glucose is a hexose, whereas a pyruvate is a triose, or a compound that is formed of 3 carbon atoms. So in order for pyruvate to undergo full oxidation, it needs one turn of Krebs cycle. So here we need one turn of Krebs cycle, and here we need another turn of Krebs cycle. So finally, we need 2 turns of Krebs cycle, meaning that Krebs cycle has to function twice. What does it mean that we need 2 turns of Krebs cycle? This means that Krebs cycle has to function twice in order for complete oxidation of a glucose molecule to occur. And what do we mean by complete oxidation? Complete extraction of the energy from the glucose molecule. Okay. Now, what are the energy products of Krebs cycle? Of course, we're going to see Krebs cycle, but let's first see the overall resultant of Krebs cycle. Krebs cycle has to turn twice for every glucose molecule. And there is direct formation of 1 ATP molecule per turn by substrate level phosphorylation, but the remainder of the energy will be stored in the hydrogen atoms that are transferred to NAD and another hydrogen carrier that is called FAD, or flavin adenine dinucleotide. This one is nicotinamide adenine dinucleotide, and that one is flavin adenine dinucleotide. So actually they get reduced. They were oxidized at the beginning, and then when hydrogen was lost from the pyruvate skeleton, they become reduced. So here we have a reduced NAD, and here we have a reduced FAD. Why do we say that the energy has been transferred to NAD? Because the electrons in those hydrogen atoms are high energy electrons. So electrons in those hydrogen atoms that are carried by the hydrogen carriers store a lot of energy, and therefore they should be accepted at a high energy level. Electrons in those hydrogen atoms are high energy electrons. Okay, this NAD and this reduced NAD and reduced FAD will carry their hydrogen with the stored energy inside to the electron transport chain. Okay, the step of converting pyruvate to acetyl-CoA, as you have seen in the diagram in that equation, you see, produces from each pyruvate produces one reduced NAD. Therefore, from the two pyruvate, it will produce two reduced NAD. So the conversion of pyruvate to acetyl-CoA, and that's why we call it oxidative decarboxylation, because there was oxidation of pyruvic acid, and since oxidation is always combined with reduction, then there has been reduction of reduced NAD. If you look at the energy yield of Krebs cycle, you will see that the overall will be the 1 ATP that has been formed by direct phosphorylation, the one we have talked about, and 3 molecules of reduced NAD. So there was enough hydrogen removed. There was enough hydrogen removed from glucose in order to reduce 3 molecules of NAD and 1 molecule of fat, in addition to carbon dioxide. Of course, this carbon dioxide will be later on exhaled, okay? This is the yield of 1 turn of Krebs cycle. So the yield of 1 turn of Krebs cycle is 3 molecules of reduced NAD, 1 molecule of reduced fat, and 1 molecule of ATP. This 1 molecule of ATP has been formed by direct phosphorylation or substrate level phosphorylation, but this time in the matrix of the mitochondrion. So we have seen two examples of substrate level phosphorylation. The formation of ATP in the cytosol by substrate level phosphorylation, and the formation of ATP in the mitochondrial matrix that is more or less similar to the cytosol, okay? So, of course, this ATP, this direct ATP, is a very small amount of energy. Is that all the energy that has been obtained from Krebs cycle? Of course not, because we have a lot of indirect, invisible energy that is stored in reduced NAD and reduced fat. Why? Because these 2 reduced molecules will go to the inner mitochondrial membrane and will deliver their hydrogen atoms to the inner mitochondrial membrane. And the electrons on those hydrogen atoms will be transferred from one electron carrier to another with the release of energy that will form all the ATP that is missing to complete the total number, which is 38 ATPs. How does this occur? What's the energy yield of reduced NAD and reduced NAD? Every molecule, we are going to write this on a new screen because it is important. So on this screen, we are going to discuss the indirect energy yield of Krebs cycle. The indirect energy yield of Krebs cycle. So if you have 3 molecules of reduced NAD and you have 1 molecule of reduced fat, and if you know that every molecule of reduced NAD will give you 3 ATPs along the electron transfer chain, then the total energy yield of those 3 reduced NAD molecules will be 3 by 3 or 9 ATP molecules. And if you know that every reduced fat molecule along the electron transport chain will give 2 ATP molecules, therefore, the total energy yield will be 1 by 2, which is 2 ATPs. All this energy yield, which is 11 molecules of ATPs per turn of Krebs cycle. This is what we call the indirect energy yield of Krebs cycle. This is what we call the indirect energy yield of Krebs cycle, the indirect energy yield of Krebs cycle, okay? If you add to this the one that is produced from direct phosphorylation, then we would obtain the total energy yield of Krebs cycle. So the total energy yield of Krebs, the total energy yield of Krebs cycle would be the direct energy yield plus the indirect energy yield, the direct energy yield plus the indirect energy yield. So the direct energy yield is 1 ATP per turn. The indirect energy yield is 11 ATPs per turn. So the total energy yield of Krebs cycle is 12 ATPs per turn. Okay, this is 12 ATPs per turn. So the total energy yield of Krebs cycle is 12 ATPs per turn. And now we move on to something else, which is like, what is the energy yield, from one glucose molecule, in Krebs cycle, in order to answer this, we have to remember that one glucose molecule gives 2 pyruvate via glycolysis. So one glucose molecule gives what? Gives 2 pyruvate via glycolysis. And those 2 pyruvates require 2 turns of Krebs cycle. And therefore, the energy yield of the 2 turns, the energy yield of the 2 turns equals 12 multiplied by 2, which is 24 ATPs. But of course, we have to add here the total energy yield, and here we have to add the total energy yield, okay? And this is the citric acid cycle. This is the first reaction of the citric acid cycle. Acetyl-CoA from oxidative decarboxylation, of pyruvic acid combines with oxaloacetic acid in order to here is acetyl-CoA, and it combines with oxaloacetic acid. So actually the beginning is here. This is reaction number 1. And then this is the first product. So actually this is the first product. I'll erase because it's too messy. So here, this is the first chemical reaction, which is combination between acetyl-CoA and oxaloacetate in order to form citrate, and then citrate is the first product. It gives isocitrate, which is the isomer, and then carbon dioxide will be removed. Of course, here, this one consists of 2 carbon atoms, that one consists of 4 carbon atoms, so citric acid is a 6 carbon atom. Since this is the isomer, then it will also consist of 6 carbon atoms. since you removed carbon here, then alpha ketoglutarate will consist of 5 carbon atoms. Another carbon atom is removed here in carbon dioxide. So succinyl coenzyme A consists of 4 carbon atoms. Here, all what we have done is to add phosphate, and the 4-succinate also consists of 4 carbon atoms. Since we didn't remove any carbon dioxide, then the number of carbon atoms will remain the same. It's worth noting that we have 3 reduced NAD molecules that have resulted and 1 reduced FAD molecule, as well as 2 carbon dioxide molecules. Notice that for the total number of carbon atoms that are produced in cellular respiration, we are going to open a screen and say, we used to say that when we have a glucose molecule that binds with 6 oxygen molecules, it will give rise to 6 carbon dioxide, 6 molecules of water, plus, of course, here I should have written carbon dioxide, and here I should have written water, and here we have the 38 ATPs. And now let's calculate how the 6 carbon atoms have been obtained. So the aim of that screen is to show you how the 6 carbon atoms were obtained from 1 glucose molecule. If we move back to the pyruvate reaction, we will find that per pyruvic acid molecule, there is 1 carbon dioxide that has been removed by oxidative decarboxylation. So actually, since I moved backward, I have to open a new screen again. Okay, so here, 1 glucose molecule upon complete oxidation under aerobic conditions, is supposed to give you 6 carbon dioxide molecules. 6 carbon dioxide molecules. How did we obtain the 6 carbon atom molecules? We have 1 glucose, and that glucose through glycolysis will produce 2 pyruvate molecules. The 2 pyruvate molecules through oxidative decarboxylation, as they are entering the mitochondrion, will lose 2 carbon dioxide, okay? And then they will join Krebs cycle. Every turn of Krebs cycle forms or gives rise to 2 carbon dioxide. So we have 2 carbon dioxide molecules per turn. So we have 4 carbon dioxide molecules per 2 turns. So here we obtained carbon dioxide. Here we got 2 carbon dioxide, and here the final through Krebs cycle is 4. Then the resultant is 6 carbon dioxide. Part of this carbon dioxide will be used in the human body for buffering systems, for example, and or for carboxylation of another substrate, and part of it will be exhaled through air. Here, of course, as we said, NAD is nicotinamide adenine dinucleotide, and a nucleotide would consist of adenine plus a ribose sugar plus a phosphate, and that's why we call it nicotinamide adenine dinucleotide. FAD is flavin adenine dinucleotide. Both are coenzymes that are required, but here, of course, I'm sorry, there is no phosphate. So actually, nicotinamide is a vitamin, and that you have adenine, which is a nitrogenous base, and since the structure is very close to the nucleotide, then we call it nicotinamide adenine dinucleotide, and here we have flavin adenine dinucleotide. They are both coenzymes. Their function is to carry protons and electrons. Actually, protons and electrons are Yani one proton with one electron is nothing but the hydrogen atom. Any hydrogen atom that is removed from glycolysis, as I told you at the beginning of respiration, as well as any hydrogen atom that is removed through the citric acid cycle, will have to be carried on a hydrogen carrier. Nicotinamide, adenine dinucleotide, and FAD are considered hydrogen carriers. We can call them hydrogen carriers. We can call them electron carriers, because the hydrogen atom, the proton will separate from the hydrogen at the electron transfer chain, and we can call them coenzymes. Why do we call them coenzymes? They are large organic molecules. As you know, coenzymes are large organic molecules that speed up chemical reactions and help activate enzymes by making their active site more fitting with the substrate. And they are important because if they are not there, hydrogen wouldn't be carried or trapped by them, and therefore it would return to the carbon skeleton. What we say that they are coenzymes, so they are enzyme helpers. NAD is a coenzyme for an enzyme called NAD dehydrogenase. FAD is a coenzyme for an enzyme called FAD dehydrogenase. A dehydrogenase, as you know, the prefix D means removal, and therefore the dehydrogenase enzyme, D means removal, okay? So both enzymes catalyze removal of hydrogen, meaning that both enzymes catalyze oxidation, removal of hydrogen from the substrate, which is glucose. But in NAD dehydrogenase, that hydrogen will be transferred from glucose to NAD. So NAD will be so glucose will be oxidized and NAD will be reduced. In the FAD dehydrogenase enzyme, the hydrogen atoms will be again removed from the carbon skeleton of glucose and be transferred to FAD. So the carbon skeleton of glucose will be oxidized, whereas FAD will be reduced. NAD and FAD are vitamin derivatives. And as I said, without NAD to accept the protons and electrons, glycolysis and Krebs cycle would stop and the whole cell would die because it would be deprived of energy. So the cause of death here would be the deprivation of energy. Okay, they are vitamin derivatives. Nicotinamide is niacin, and niacin is vitamin B3, whereas riboflavin is vitamin B2. I don't know why I wrote riboflavin here. So nicotinamide actually is niacin or vitamin B3. As we said, NAD plus is the oxidized form. NADH plus, the proper, you will see this written in many textbooks, but the proper way to write it, as we said, is NADH plus. This is the reduced form of NAD, and you can just write NAD and Re. It's clear that fat carries the two hydrogen atoms as a whole, meaning that it carries two protons and two electrons. But for NAD, it's different because NAD will carry one proton and one whole hydrogen atom only, meaning that NAD will carry this. We said that if NAD that carries a positive charge meets two hydrogen atoms like this, two protons and two electrons, then it will carry, this electron will neutralize the positive charge. So it will carry one proton, which is this one proton, okay, and actually two electrons. If we count the number of protons. Okay. And now we move on to the process of oxidative phosphorylation and the electron transport chain, but we will be doing this on a separate session or in a separate session, inshallah. Thank you guys, and see you on Thursday.